So good evening to one and all present here. So we will see today week 8 live interaction session on the NPTEL course Environmental Biotechnology for the NPTEL course ID NOC 25 BT 83. I hope my slide and voices, my slides are visible and the voice is audible. I hope.
Yes, sir.
Okay, then we will move on to this today's session. So today's session, as usual, it is going to be first the recap of the week 8 lectures, followed by the important MCQs, which is sample MCQs for the environmental biotechnology course. So myself, Ajit Kumar, a PhD research scholar from Department of Bioscience and Bioengineering from IIT Guwahati. Myself, Ajit Kumar, a PhD research scholar from the Department of Bioscience and Bioengineering from IIT Guwahati. So moving on to the today's lecture, before moving on to the today's lecture, I will introduce myself. Myself, Ajit Kumar, an JDP, Joint Degree Research Scholar, which is a research collaboration between IIT Guwahati and IIT BHU Varanasi. So I work in a laboratory of biochemical and environmental engineering, where our lab focuses various wastewater treatment problems, which has been seen in the different industries. Especially my research inclines in the application of enzymes in the wastewater treatment, especially in the textile wastewater treatment. Then moving on to my qualification, I have done BTech Biotechnology from Shastra University and MTech Industrial Biotechnology from NITK Surathkal. So moving on to my work experience, I have worked in several pharma companies like Biocon and Enzyme Bioscience. This is all about my personal introduction. So now let's dive into the week 8 lecture recap. So before moving on to that, in the last class, we have seen some important concepts, which is method of microbial ecology. We have seen several methods where we have discussed one anomaly, which is great plate count anomaly. Followed by we have also seen categories of culturable organism, where there are two broad categories, which is cultured organism and cultured organism. Where in uncultured we have seen culturable and non-culturable organism. And also we have seen the culture dependent methods, especially we have deeply discussed on direct culture and enrichment culture technique and what are all the selection pressure that one need to be considered during the enrichment culture technique. And also we have seen other alternative methods for culture dependent methods for the isolation of any microorganism from the environmental sample. Then we move on to the culture independent method, where we have discussed the staining and the microscopy technique, where first we seen the general staining procedures and then we move slowly on to the viability staining approach, where we have seen how to differentiate the live and dead cells using different dyes. And we have also seen the green fluorescent protein and what are their advantage and how it is used for microscopy. And we have seen some of the limitation of general microscopy. And we have seen advanced technique like fluorescent in situ hybridization technique. And we have discussed their advantage and we have seen the types of fish, which is fluorescent in situ hybridization. So these are all the content that we have gone through in the last week live interaction session. And I hope you have also gone through this week's live lectures. And I hope you have all finished the assignments on time. So now we will see in this lecture the week 8 recap, where first we will see what is the summary of the culture independent methods. As we have already discussed the staining and microscopy technique in the earlier, if you have missed out, you can go and rewatch it. It is available in the YouTube. And if you are not able to find it, you can also find it in the NPTEL environmental course section where there is a separate tab which is recording sheet where you can access both the PPTs and lecture videos. So last class we have discussed the staining and microscopy approach, and what we have seen traditionally the microbiologists they try to rely on growing microbe in the lab to study them. But we understand, and we are from our past lectures, we understand that 99% of the microbes in nature cannot be cultured. Then how do we study them? We can study them by using appropriate culture independent techniques. And we will see what are all the culture independent techniques one can follow in order to understand the microbial diversity. And first, we have discussed as staining and microscopy, we have discussed the advanced techniques like fish also. And in today's class, we will be more focusing on the specific gene based approaches. And if you see the specific gene based approaches, you have two important markers which are very important to identify any bacteria or any microorganism in the environmental sample. So if you see the taxonomic marker, the meaning of taxonomic marker you can correlate with uh, when we go to supermarket, everything as a barcode, right? What is barcode? When we scan these barcodes, we could get all the information about the thing that we are going to purchase. Likewise, the microorganism, every microorganism has a barcode. What is barcode in the sense? That is a specific gene, which is 16S rRNA gene. If in the case of bacteria, or if you are focusing on eukaryotic or fungi microorganism, then you can go for 18S rRNA or ITS sequencing. So these are all like specific conserved regions which helps us to identify the microbes that are present in any environmental sample. So it is very important to understand the taxonomic marker, that is a 16S rRNA if it in case of prokaryotes, and it is if it is eukaryotes you can go for 18S rRNA. So these are called taxonomic marker. It helps to identify the microorganism that are present in the sample. So if you want to understand the functional gene and their metabolic activity, then you can focus on functional markers. In functional markers, what do you mean that it not only tells who is there, that is, what are all the microorganisms that are present in a given environment, but it also tells what they can do. That is, what really they are capable of doing in that environment. So it answers both the questions. Whereas in taxonomic, you can only get the answer who is there in the given ecosystem. So here we could get what they can do in the particular environment. So how do we find them? By using the quantitative PCR, which helps us to measure the relative abundance of the microbes in the sample. So these are all the two important markers, which is taxonomic and functional marker, which helps us to identify the microorganism without even culturing them in an appropriate environment, appropriate culture. So if we see in terms of environmental genomics, these specific gene-based approach helps us to understand the community profiling. Community profiling means we can understand who are all present, what are all the microorganisms that are present naturally in the environment by using the above technique, and also we can identify or we can evaluate the proportion or abundance of the microorganism in the environment. And we can also go for whole genome, metagenome analysis where we can analyze all the genetic material information about a microbial community. So these are all the uses of these specific gene-based approaches with regard to the environmental genomics. So we will see different culture independent techniques in the upcoming slide, and we will see one by one. Moving on to the techniques first we will give a broad overview of how culture independent technique is followed. First, what we will do? First, we will be collecting the sample. It can be either environmental sample, such as soil sample, water sample, or wastewater sample. So first, we will transport them in the laboratory using some sample containers. After transporting, first step is to extract the total community DNA or RNA. It can be a DNA or RNA depending upon the technique and what are all the information that you need. So depending upon you can extract the total DNA or RNA. After successful extraction of total DNA or RNA, your next step is to amplify this DNA by using appropriate PCR technique. And we all know that PCR is nothing but polymerized chain reaction. So this helps us to amplify the gene sequence that is available in the total DNA. So once you have amplified, the your next step is to either you can go for community profiling to know what are all the microorganisms that are present in the environment, or you can know how much abundance or prevalence of microorganism. For as we have seen many examples in the previous lectures, for example, I will repeat it once. So if you are focusing on a hot temperature region, you will expect thermophilic microorganism as a dominant species compared to the mesophilic or saccharophilic microorganism, right? So you could identify the relevance or abundance of the microorganism as well as you can identify what are all the different microorganisms that are present in the environment by using the various culture independent techniques. So this is the general procedure that is followed for the culture independent technique. And we will move on to techniques, and these are all the some of the common techniques, common DNA fingerprinting methods that are available for culture independent method, where the first method amplified ribosomal DNA restriction analysis, and second one is terminal restriction fragment length polymorphism, and third is amplified ribosomal intergenic spacer analysis, and fourth one is denaturing gradient gel electrophoresis. So these are all some of the methods that are available for identifying the microorganism without even culturing them. But in this lecture, we will focus only on the first and fourth one, which is amplified ribosomal DNA restriction analysis, ARDRA, and denaturing gradient gel electrophoresis. So we will see them in the upcoming slides. And before that, you have to know the term phylotype because it is very important while we are trying to describe the microbial diversity of the habitat based solely on the nucleic acid sequences. So this term refers to the microbial diversity in a particular habitat, which are solely classified based on the nucleic acid sequence. And it is only when additional physiological and genetic informations are became available. Typically, what usually happens after the microorganisms are brought into the laboratory condition, which helps us to identify their genus and species name for a phylotype, right? So before moving on to the each techniques, you have to understand the term phylotype. Then we will move on to the first technique, which is amplified ribosomal DNA restriction analysis. And the term itself describes that it is helps us to identify and classify them by analyzing their DNA sequence by using restriction enzymes. So now we will see how it is done. First, as usual, you have to first start with the environmental sample. It can be any environmental sample, water, wastewater, or soil sample. And the first step, you have to extract all the DNA together out from the environmental sample. Then you use polymerized chain reaction to amplify the 16S rRNA gene, which is a taxonomic marker, which is regarded as a universal primer or taxonomic group specific marker. So it acts as like a unique ID to every microorganism. Once you have amplified the 16S rRNA gene, you have to confirm through agarose gel electrophoresis. Then the PCR amplified product, you have to, this PCR amplified product, which is gene, it has to be integrated into the vector. So the next step is cloning these 16S rRNA gene into a appropriate vector. Then you have to create the library, which is a clone library. After creating the clone library, you have to analyze the positive clones for 16S rRNA gene insert by appropriately using restriction enzymes. What is the role of this restriction enzymes? It cuts the DNA at the specific site, producing unique binding pattern. So, by analyzing these binding patterns, which allows the researcher to group the microorganism, which allows us the researcher to group the microorganism into different types. For example, micro group 1, group 2, group 3, group 4, where group 1 can survive in the hyperthermophilic condition, whereas group B, it can survive in the mesophilic condition. So first, scientists will try to segregate the microbes into different types based on the pattern that they got after restriction digestion. So this is how they group the microorganism and try to identify the microorganism whenever we give an environmental sample. And this is one of the powerful techniques that has now being used to identify the microbial diversity in any environment. So this is regarding the first technique, which is important and most powerful, which is the amplified ribosomal DNA restriction analysis, where the scientist uses restriction enzymes in order to find a particular binding pattern. And the next important technique is denaturing gradient gel electrophoresis, which is DGGE. This technique, before moving on to technique, first we will give one situation where that all DNA may look similar in length, but they have differences in base pair. Right, each DNA has different base pair. Then how do we tell these nearly identical sequence? How do we segregate them and identify them? In that case, if you are trying to separate very similar genes, you can use this technique, which is denaturing gradient gel electrophoresis technique, where it tries to separate the genes based on the melting profile of the each DNA, because every DNA has different melting profile, and we will see the melting temperature concept in the upcoming slide. So the first most important principle, the basic principle of this technique is they try to separate the genes of the same size that differ in their melting profile, because every DNA has their different gene sequence. So, as I said, the separation is purely based on the melting behavior of the double-stranded DNA. And melting behavior, what it depends upon? It depends upon the base pairing composition of the DNA. And in a DNA, what pairs with what? Adenine will pair with adenine will pair with always thymine, which is A will pair with T and C will always pair with G. And what is the hydrogen bond between them? Between A and T? It is double bond or triple bond?
It is double bond.
Yes, it is correct. It is double bond. Whereas between C and G, it is,
Triple bond.
yes. So among these two, which is more difficult to break, the AT bond or CG bond? Anyone? So as you told, the A always pairs with T and it is linked by double bond, whereas C always pairs with G with triple bond. So which is very difficult to break when we are subjecting them to the temperature. So it is obviously it is which is having the triple bond. as you need to spend higher amount of energy when compared to breaking the ANT base pair. So this is all about the base pairing composition, and this is the one of the main important factor when we are dealing with the melting behavior of the DNA. So the GC clamp, which ensures that at least a part of the DNA remains double stranded. So we will increase the temperature, or we will try to use some denaturant. We will use some denaturant to the DNA which is double stranded, and we will weaken the zipper. That is, we will try to break the AT bond and opens up while the GC base pair zip stronger. So we will increase the temperature to the level that GC will be still in the DNA. So this is necessary so that separation on the polyacrylamide gel with a gradient of increasing concentration of denaturant. The denaturant can be a formaldehyde or urea. So we will check the concentration of the denaturant so that the GC clamp will remain intact. So what happens if this GC clamp is absent? The DNA would finally denature into single strand. So that we cannot amplify the DNA out of it if it is become single stranded. So the main principle of DGE is here we are trying to separate them based on the melting behavior, and we will run the polyacrylamide shell by using denaturant or temperature. So now we will see the concept of melting temperature in a DNA. So if you see the DNA, as I said earlier, the double-stranded DNA where A always pairs with T and it is double bond. Whereas in CNG it is triple bond. So as we increase the temperature at one particular point, we call it as a melting temperature. Melting temperature, where you could see the A and T bonds are break where the GC is still intact. We call this TM, which is melting temperature. So above that, what happens? Above that, the double stranded will be cleaved into single strands. So we have to find first the melting temperature of each DNA. Then we will separate each one based on the band that we are observing. So first, the main principle is denaturation of DNA by either we can use the temperature as one of the factor, or you can use denaturant. As we have seen in the earlier slide, that denaturant can be formaldehyde or urea. So if you increase the melting temperature, the GC bond breaks. So here the main function is you have to retain the GC clamp, and if you analyze them on the polyacrylamide gel, which is containing the increasing gradient of denaturant. For each of this DNA, which is double stranded, you could observe there will be a transition from the double stranded to the single stranded secondary structure at the central position in the gel. And also, this will stop or at least strongly slow down the migration of the respective gene frag. So the main principle here is the melting point where we will use some denaturant or heating for identifying the microorganism. So so if you see from the principle, we could understand the importance of GC rich region of the PCR primer, which acts as a clamp to prevent them forming the single stranded DNA. And the result of this analysis of the PCR product obtained from the micro community is the band pattern, and number of band is the rough estimate of microbial diversity that are present in the given ecosystem. So when we will apply this technique we will apply if two DNA sequence have the same size. If they are having the same size and when it is very difficult to separate them, you can use this DGE method where the separation will be more solely based on the melting temperature, because every DNA has the different gene sequence. So their DGG separates because of their melting differences. And what ensures this DNA doesn't fall apart? Here only the GC clamp comes in. So it acts as a safety pin at the end of the zipper. So after analyzing this band pattern, what are all the information you can get from the DGE band product? So first you can analyze the complexity of the community, how different microbes are present in the given ecosystem. Then you can identify the community member by going further sequencing using advanced sequencing methods. Then you can also understand the distribution of the microbial population inhabiting different environment. And moreoverly, in the real time, you can analyze the changes that are happening in the microbial community. So these are all the some of the informations that you can get from the DGE. So if you see the application point of view, the main advantage of this method is it's more reliable, reproducible and rapid, and it is inexpensive when compared to the other advanced method. And more importantly, multiple samples can be analyzed simultaneously. So this it is making possible to follow the changes in the microbial population. For example, if you take a if you want to analyze the seasonal changes of the microbial community in an ecosystem is at that point you can use DGGE because multiple samples can be analyzed concurrently. And it is particularly useful to detect the population changes, addressing the question of stability and how it changes its dynamics of the community that are present in the ecosystem. And if you see the rDNA based DGE pattern, it is solely determined by the presence of DNA. And therefore, in first approximation, based on the band pattern, you could say how much abundance of populations are there in the environment. And it should be more strongly represent if you are going for a rDNA based approach. So if you are going for a DNA based DGE pattern, you could get the information of population abundance. And if you are going for rDNA based pattern, here you could get the information about the genome, genes, what are all the genes which are metabolically active. So by using the both, whether you are extracting the DNA or RNA, depending upon the sample, you can analyze different inference out of it. So this too is the one of the important method, and we will see the few more advanced sequencing techniques. The first technique is next gen sequencing technique. So what happens in the earlier days? In earlier days, in order to understand the microbial diversity, they rely on constructing the clone libraries to separately amplify the DNA molecules, and we call this amplified DNA molecules as amplicons. So what happens? Scientists used to separate each DNA, and they will put each DNA into a separate cloning vector, and they will try to amplify them. Then they will go for sequencing. So this a single step will even takes days of time, and it is very costly too. So each clone in the library contain a unique sequence. So that will be used as a template for sequence determination. So this earlierly this used to take a large amount of time, and we have to spend more amount of money also. So what happens frequently? These approaches target DNA, allowing the researcher to study the taxonomic and functional diversity of the sample, regardless of whether DNA is originated from dead, alive, metabolically active, or dormant organism. So earlier they used to target only DNA sequence. They target DNA sequence, and the researcher will try to study their taxonomic as well as the metabolic function. But now instead of RNA, DNA, they are also using the RNA as a template and trying to study their uh, genes which are metabolically active. So, by analyzing the RNA, Scientists are now trying to understand the both taxonomic and functional diversity more detail, and these RNA-based technique also has some challenges when compared to the DNA-based techniques because these RNA are generally unstable and their extraction efficiency from the sample is very low. So these are all the two important reasons why scientists are going for DNA-based methods more often when compared to the RNA. But if your goal is to study the uh, metabolically metabolically active metabolically active gene, then you don't have any other option. Then at that case, you have to go for RNA-based sequencing approach. But you have to remember these RNAs are often unstable when compared to DNA, and the extraction efficiency is also lower when compared to DNA. So, as earlier, scientists were trying to clone them individually, and this causes loss of time and also money. So that's where the next generation sequencing has come into play, and this next generation sequencing is high throughput sequencing where at a time you can analyze billions of nucleic acid fragments, and it can be analyzed simultaneously as well as independently. And in the next generation sequencing also earlier they used to follow a classical method which is Sanger sequencing, which is also known as D-deoxynucleotide chain termination sequencing. What happens here in Sanger sequencing? They used to process one nucleotide sequence per reaction. So at a time you can analyze only one nucleotide sequence. So uh, it is very slow, but they are reliable at that stage. But as the technologies evolves, lot of next gen sequencing has come into play where parallelly billions of sequences can be analyzed within a very few amount of time. And if you see this human genome, it took more than 10 years and it is very cost effective when they are going for Sanger sequencing. But nowadays, the entire genome using the next gen sequencing can be sequenced within a single day. So earlier, what it is to be, earlier they took more than 10 years to sequence an entire human genome, but now it can be handled within one day. So the Sanger sequencing technology, we call it as first gen sequencing. It is used to decipher the human genome required over a decade to deliver the final draft. So here we compared Sanger sequencing versus the next gen sequencing, where you can think it of like an Sanger sequencing is riding a bicycle, and riding a bicycle which is slow but it is steady, but we can rely on. So you could say that in Sanger sequencing, you can sequence about 500 to 1000 base pair, but it is always time consuming and expensive. But you can relate the next gen sequencing to like car or rocket. So what happens here? Here we could get billions of data within a few minutes. So that is where the next gen sequencing has been developed. And if you see, to sequence a human genome, they took about 10 years to complete the project, whereas with the help of the next gen sequencing, they have completed within single day due to the advancement in the technologies. And, and if you see the evolution of the sequencing, in the first generation, as I said, first Sanger sequencing, Maxam and Gilbert Sanger chain termination. As I said, you can think it of like a bicycle where they are slow, steady, but they are reliable. What is the principle they use? They use GNTP to infer the nucleotide, but one nucleotide can be done for per reaction. They are relatively slow and expensive, where you can analyze 500 to 1000 base pair fragments. So for next, as the technologies evolves, the next generation, which is second generation, we often call it as next gen sequencing, where different techniques like 454, Solexa, Iron Torrent, and Illumina came, where millions of fragments has been analyzed within few minutes. So here you can correlate this with a like a car. When compared to bicycle, car is much faster, right? So it has higher accuracy of predicting the nucleotide, and also it can sequence millions of fragments, and it is more faster, and it is very cheap and more accurate too. So these two are for short read sequencing. That is, if you are want to read a very short read sequencing, like up to 1000 base pair, you can go for second generation sequencing. Like you can use the technology like Illumina and Torrent or Selexa or 424. Suppose if you have a very long DNA sequence, that is more than a KB, that is 1000, 2000, 3000, then you can go for third generation, where you can think of like a rocket or aeroplane, where it reads a longer piece of DNA in the real time within a few minutes. So it can sequence high amount of DNA, which is a long reading sequence and makes cost effective. So this is how the sequencing evolves during the course of time. And now second and third generations are more famous. And if you're focusing on short read sequencing, then you can go for second generation. And if your read is long read sequence, then you can go for third generation sequence. So these are all the evolution of sequence. And if you see this gene sequencing in terms of microbial community analysis, the rapid growth of this sequencing technology and due to the computational power over the last 10 years, it has transformed the field of molecular ecology. And in previous slide also, we could infer that millions of base pairs can be sequenced within a few minutes. So the use of traditional clone libraries when coupled with the first gen sequencing technology like Sanger, what happened? It allowed only few number of amplicons to be sequenced in the serial manner. That is, you can analyze one nucleotide per reaction only. So where due to technology evolvement, next generation sequencing allows us to sequence millions of amplicons within a few minutes in a parallel. So this is the major breakthrough provided by the next gen sequencing approach, is that their entire genetic soup. Genetic soup is nothing but a different DNA sequences in a microbial community. So it allows this entire genetic soup to be analyzed parallelly with help of different sequencing technology while ensuring the sample they originate from. It is also recorded, and we call this the sequencing process as amplicon sequencing or metagenetics or metabarcoding. And we will be seeing the metagenetics in the upcoming slide. So till this we understand the power of sequencing and how it is evolved over the decades. And we have seen the first generation, second generation, and third generation too. Now we will move on to the metagenetics. Metagenetics. So it is one of the very important tool for biodiversity survey. So it helps us to study the biodiversity by looking at their DNA sequence in an environment. So what happens here? First, we will collect the DNA from the sample and we will infer the what kind of microorganisms that are present in the given sample. So the first important application is there, species identification. So what kind of species that are present in the environment and their relative abundance. How much microorganisms are present based on the identical DNA sequence that has been recorded. And second is their rare biosphere. That is, many microbes exist in very small number. So we might miss them. But using metagenomics, it allows us to detect even those minute or little organisms that we have missed by the traditional approaches. And also, by targeting the specific microbial taxa, this approach effective in screening them and also identifying them. And this is why they have been used in many applications like for the contaminant removal or for pathogen detection or to study the antimicrobial resistant genes or to monitor a particular process like dissimilatory or ammonia oxidation methods. So it is used for targeting a specific microbial taxa. That is some if suppose for example, if some microorganism, you want to isolate a microorganism that needs to be degrading the arsenic, then what you will do, you will collect the sample from the arsenic contaminant site and you will try to analyze the DNA using the appropriate technology and try to find the arsenic resistant gene out of it. And if this microorganism, the microorganism that you are focusing on has the arsenic resistant gene, then you could use this microorganism for arsenic remediation. So this is where the metagenetics comes into play and it is very important for biodiversity analysis. And when this technique applied to the environmental DNA, the metagenomic can be used to quickly determine what are all the microorganism as well as the macroorganism and their abundance and their relative abundance can also be measured. So this is how the metagenomics is important. And in metagenomics, you have to understand the one most important term, which is operational taxonomic unit. First, you have to understand the term taxonomic operational taxonomic unit, where what we will do is uh, first we will collect the environmental sample, analyze or we will analyze or extract the we will analyze or extract the whole DNA out of it. Then we will segregate them into a particular group. So what happens here? So so it is like a bucket where we place all the DNA sequence that are very similar. So instead of looking at every DNA sequence, we can group them if they have at least 97% identity. So for example, here there are some 20 reads. First we will segregate the reads, and if suppose the read 1 and read 2 having the percentage identity of more than 97, we will group them into one group, that is OTU1. And the OTU2, these are all the sequence that has the 97% identity. So likewise, we will segregate them and this will help us to save the time and also eliminates the bias. So the species separation requires at least 97% identity. And why 97? So if we see, we usually separate the bacteria at the genus level, and if you want to, if you are targeting for the genus level identification, 97% is enough. But if you are targeting for a species level separation, then you can increase the threshold to 98 or 99% sequence identity. And how it works, the DNA, first you have to DNA sequence and you have to compare each DNA sequence with each other. And if two sequence having more than 97% identity, then you can group them together and each group will serve as and one ATU. So here the main important factor is clustering them and putting in an appropriate bucket so that it will be very easier for our taxonomic identity, because studying each one is very difficult. So if we apply this OTU, operational taxonomy unit, where the two sequence have more than 97% identity, so it will be easier for our analysis and we could get a clear picture of the microbial diversity. So these are all the important concepts regarding the NGS. And if you see the limitation of this 16S rRNA gene amplicon sequencing, the first disadvantage is their choice of hypervariable regions in the 16S rRNA gene, which often leads to giving different results every time. So we have to first observe and choose the appropriate hypervariable regions in the 16S rRNA gene. And second major drawback is that the taxonomic collection classification at the genus level, it is also depends upon the database and the classifiers that you are using. It is always recommended to use an uniform global database and classifier so that we could more appropriate in identifying the microorganism. And due to the limited functional information on the microbial community, which also limits the 16S rRNA gene amplicon sequencing method. Then we will study the metagenomics. As we have already seen, the metagenomics is studying the genetics of the microorganism in a given environment. So it circumvents both unculturability and genomic diversity. So it is the biggest roadblock to advance in clinical and environmental biotechnology. So that's where the computation methods as comes into play for maximum understanding of the genetic composition and the activity of the community, so complex that it can only be sampled, never completely characterized. And if you see the process of metagenomics, as we have understood, metagenomics is nothing but understanding the genetic information that are present in a microbial community. So what happens here, how genes might influence each other's activity, and also what are all the genes, that is microbes, are present in a given environment. So if you see the procedure for the metagenomics, as we already seen, first you will be collecting the sample from various source, and we will be first isolating the or extracting the total DNA. Then we will construct the DNA library. Then we will go for NGS approaches for DNA sequencing. Then after sequencing, we will analyze and reconstruct and form contact regions. And with assembled genome sequence, we will use different bioinformatics tools for analyzing the sequences. So due to the advent of next generation sequencing, it has provided an easier access to the separate approach that is not reliant on the sequencing of PCR fluid, but capable of providing the information termed metagenomics. So if you see the procedure, first, first you will be collecting the sample and processing it. That is, first you collect the sample and try to extract the total DNA out of it. After extracting the DNA, construct the metagenomic library, where you will be cloning and transforming the extracted DNA into the suitable host cell. So after transforming, you will be screening for metagenome library, where you will try to identify and isolate specific genes. Then you will go for DNA sequencing approach like NGS technologies, where we have seen lot of second and third generation sequencing, through which we will be attaining the DNA sequence. Then you will assemble the sequence. What do you mean by? You will reconstruct the genomes from the short sequencing read. So once you have got the short sequencing read, then the next step is binning, where you will group the more similar DNA sequence into a group or bins that represent individual genomes. from closely related organism in a microbial community. Then we will go for annotation, where the genes within the assembled sequence are identified and annotated to predict their function using bioinformatics tools and database. Then you will be going for experimental design and statistical tool. So here you will be using various statistical tools like metastats in order to validate the design design and last you will be storing the data and sharing the information through various centers like National Center for Biotechnology Information for sharing the information that you got through the metagenomics approach. So this is the general procedure for metagenome, and this is how the metagenome analysis is done with the help of the different statistical tools, bioinformatics tools, and if we are moving to the outcome of the metagenomic analysis, it helps us to answer all the basics four questions that we see in the microbial ecology. The first is it answers the question of who is there. That is, try to analyze all the microorganisms that are present in the given ecosystem. And the next question is what they can do. It tells us the metabolic potential of all the genes and what are they doing. It tells us the metabolic potential of the microorganism and how do they interact with themselves, also with the abiological environment. That is, how do they interact the biotic and abiotic factor. So here you can use different networks and models, and here more importantly you need to use different multiple samples and data set. As it is a complex environment, you need to consider all these, and these all information you can get through the metagenomic analysis. Then the advantage of metagenomics is that first, you can accurately identify the microorganisms since this technology, this metagenomics can read the DNA directly from the sample. And next is it tries to predict the functional profile in the community. What does it mean? It means it not only identify who is there, but also it tries to tell all other information like what can they do, and how do they interact with biotic and abiotic factors in an ecosystem. So it typically answers all the goals of the microbial ecology, and it avoids the bias of the PCR amplification. So why does it happen? So while designing the PCR with primer, sometimes you miss certain DNA sequences. So this often leads to the bias. But with metagenomics, this bias it's often reduced or it is avoided. Then less biased diversity survey can also be done through bioinformatics. So with bioinformatics and metagenomics, it can extract the most important genes, which helps us to tell about their evolution or function, such as what are all the antibiotic resistant genes are present, what are all the functional markers that are present, taxonomic markers, etc. So these are all the key points of advantage while going for metagenomics. And the last concept for the today's class is the meta-transcriptomics. And the transcription, the word itself telling us that here we are focusing on RNA. So if you are examining the RNA instead of DNA, and if you want to analyze the genes, then you can go for meta-transcriptomics, where RNA will be extracted and we will be studying the not only what microorganism could do, but what they are actively doing at that environment. So this study, we call it as meta-transcriptomics. So it examines and it provides a detailed picture of functional and metabolic activity of a community at a given moment, and it also examines instantaneous changes that happens to the environment in real time. So this is how the meta-transcriptomes works. That is, if you are focusing on a functional and metabolic activity of a community, you can go for meta-transcriptomes, where RNA will be used as a template in order to understand their metabolic activity of the microbial community. So, in addition to both metagenomics and meta-transcriptomics, alternative omics are also available, which can provide insights into the next level of biological organization. So, if you are going to study the protein that the microbes are producing, then you can go for metaproteomics, where proteins, especially it is dedicated to the proteins that are microorganisms are secreting. For example, enzymes that are breaking down the plastics in the soil. If you are focusing on plastic degrading microorganism in an environment, then you have to identify the enzymes that are capable of breaking the plastic. So you have to understand the proteins, that is enzymes first, where you can use the metaproteomics tool. And if you want to study the metabolite, which is normally the end product of any microbial community. So this study can be done using the metabolomics, where the metabolic end product of the microbial community can be studied. For example, the short chain fatty acid in our gut that directly affects the health, right? So short chain fatty acid is our end metabolite. So this can be studied using the metabolomics. So from today's lecture, we understand that the DNA can tell us the blueprint, but RNA shows the plan of what being need to be read and what tools need to be used, and if metabolites are the final products, what needs to be used. So first is, if you are need to know the functional and metabolic activity of a community, you can go for meta-transcriptomics. If you want to know genetic information of all the microorganisms in a community, you can go for metagenomics. But if your interest is focusing on the proteins that the microorganisms are secreting, then you can go for metaproteomics. No, my aim is to analyze the final metabolites that are present in the community, means you can go for metabolomics. So these are all the omics studies that are very important for analyzing the microbial ecology community. So this is all for the today's lecture. Here we have discussed many important terms, especially we have focused on culture independent technique, where first we have seen the amplified ribosomal DNA restriction analysis. Where we have seen the amplified ribosomal DNA restriction analysis and other technique, denaturing gradient gel electrophoresis. Then we have seen the next generation sequencing, where we have discussed various traditional sequencing like Sanger sequencing. Then we have seen the evolution, first gen, second gen, and third gen, and how it is correlated with the microbial community analysis. And we have seen the metagenetics, what are their advantage, and how it is done, what is the procedure for metagenomics, and what are the major outcomes and key points. And we have seen the meta-transcriptomics, metaproteomics, and metabolomics. And also we have seen the limitation of 16S rRNA amplicon sequencing also. So these are all the culture These are all the culture independent technique that are available in the microbial community studies. So if you have any doubts till now, if you have any doubts, you can post your doubts here, or otherwise we can move on to the sample MCQs for this lecture. Can we move on? So for MCQ, first you can go through the questions and you can unmute your mic and you can say the answer, or if you're not comfortable, you can post your answers in the chat box. So this MCQ would be very beneficial in helping understanding the concept much more better. So can we move on to the MCQs? Okay. So I will move on to the first MCQ. First question is the most probable number technique is used for option A, enrichment of specific microorganism, option B, dilution of specific microorganism, option C, enhancing the growth of specific microorganism, option D, estimating population size of microorganism, option E, estimating number of species of microorganism. So among this, what describes the most probable number? Most probable number technique is used for. You can post your answers. Most probable number technique is used for option A, enrichment of specific microorganism, option B, dilution of specific microorganism, option C, enhancing the growth of specific microorganism, option D, estimating the population size of microorganism, option E, estimating the number of species of microorganism. Option D. Okay. Estimating the population size of microorganism. Let's see the answer. Yes, it is correct. Option D, estimating the population size of microorganism. We will see the detailed solution now. So first, we have to understand what is MPN. MPN is the method which uses serial dilution of a sample, then observe the presence or absence of microbial growth in each dilution, which allows for the estimation of population size based on the dilution where growth is detected. So by this method, we can estimate the bacterial population size. What we normally use, first we will take the environmental sample and we will serially dilute it. And we will dilute it until no growth is observed. For example, 10 to the power of minus 5, if we are not observing any growth, then 10 to the power of minus 4 is the most probable number. So it helps us to estimate the microbial population size. And why not the other options? Enrichment of specific microorganism. It can be used in conjunction with the environment technique to isolate, but the most primary purpose of most probable number is estimating their population size of microorganism. Option B, diluting the specific microorganism. So it involves diluting the samples to determine the microbial concentration, but it doesn't specifically dilute specific microorganisms that are present in the sample. So it cannot dilute specific microorganism, it is general. It can dilute the whole microorganism. And option C, enhancing the growth of specific microorganism. If you want to focus on enhancing the growth of specific microorganism, we have a separate technique called enrichment culture technique. So it doesn't correlate with the most probable number. And option E, estimating the number of species of microorganism. So it doesn't estimate the number of different species. It can only tell the population size of the microorganism. So the correct option is option D, estimating the population size of microorganism. So we will move on to the next question, which is which of the following statement comparing the direct and enrichment based isolation of bacteria is incorrect? So among this statement, comparing direct and enrichment based isolation of bacteria, which is incorrect. Option A, direct culture increases the chance of getting the desired subgroup of microorganism. Option B, direct culturing reduces the bias introduced by the enrichment technique. Option C, the lab weeds can outgrow desired groups more easily during the direct culture. Option D, different enrichment media would be required for every distinct target subgroup. And option E, isolating the pure culture from enrichment culture will lead to loss of at least some microbial species from the culture. So among this statement, which is incorrect in comparing the direct and enrichment based isolation of bacteria? You can post your answers in the chat box. Option A, direct culture increases the chance of getting the desired subgroup of microorganism. Option B, direct culturing reduces the bias introduced by the enrichment technique. Option C, lab beets can outgrow desired group more easily during the direct culture. Option D, different enrichment media would be required for every distinct target subgroup. And option E, isolating the pure culture from the enrichment culture will lead to the loss of at least some microbial species from the culture. So which is incorrect? You can post your answers. Option A. Okay. Direct culture increases the chance of getting the desired subgroup of microorganisms. Now we will see the answer. Answer is yes, it is correct. Option A, direct culture increases the chance of getting the desired subgroup of microorganisms. This is wrong because enrichment culture increases the chance of getting the desired subgroup of microorganisms. Why it is so? Because here we will be trying to give the specific condition and the microbial nutrients to the microorganism in order to selectively select the subgroup of microorganism. That is why the option A is incorrect, where enrichment culture increases the chance of getting the desired group of microorganisms. Whereas the option B, direct culturing, reduces the bias introduced by the enrichment culture technique. Yes, it is true because direct culture, while it cannot completely remove the bias, but avoids the selection bias by exposing the original community to range of conditions. And option C, lab beats can outgrow the desired groups more easily during the direct culture. Yes, it is true because lab beats are fast growing beats. It can quickly outgrow when compared to slow growing microorganism during the direct culture. And option D, the different enrichment media would be required for every distinct target subgroup. Yes, to isolate different microbial subgroup, each with unique requirement. Different enrichment media would be required in order to selectively select the target subgroup. And option E, isolating the pure culture from the enrichment culture will lead to loss of at least some microbial species from the culture. It is also true because only targeted microbial group will be propagated, leading to loss of other microbial species that are present in the sample. So among these, option A is incorrect, and we will move on to the next question. So the next question is, when setting up an enrichment culture for microorganism capable of biodegrading the phenol, which one of the following step is most important? So I will repeat the question. When setting up an enrichment culture for microorganism capable of biodegrading the phenol, which one of the following step is most important? Option A, eliminating all carbon source except phenol from the media. Option B, eliminating all nitrogen source except nitrogen gas from the media. Option C, determine the optimal pH for phenol degradation. And option D, determine the optimal temperature for phenol degradation. Option E, determine the ideal growth condition to promote syntrophic association. So I will repeat the question. When setting up an enrichment culture for microorganisms that are capable of biodegrading the phenols, which one of the following step is most important? You can post your answers. So answer is option A. Okay, eliminating all carbon source except phenol. So now we will see the answer. Yes, it is correct. Option A is correct, eliminating all carbon source except phenol from the media. So if you see, the phenol degradation is unlikely to take place if microorganisms have an alternative carbon source, which may be easier to metabolize. That is, for example, if the microorganism has glucose or sucrose as well as phenol, the microorganism always prefer more assimilated substrates like glucose, sucrose rather than the phenol. So here you have to remove all the carbon source and you have to give only phenol as a carbon source. By which what happens here is the microorganism will able to adapt to the environment where there is only phenol is present. So in future the microorganism will be able to assimilate the phenol and degrade them completely. So that is why we have to eliminate all the carbon source except phenol from the media so that microorganism will be adapting to the environment very well. So if you see the option B, limited nitrogen restricts the growth as not all microorganism can fix the nitrogen assimilation. So this is also important but not as important as the phenol degrading bacteria isolation. So option C and D, it is related to optimal pH and temperature for phenol degradation. Yes, it is important, but it is secondary to the carbon source selection. And and if you see the temperature, it is obviously it is going to affect the growth, but it is less critical than the sole carbon source. And option E, determine the ideal growth condition to promote the syntrophic association. What happens here? The syntrophy means it is an multiple organism that works together to degrade the complex substrates. So the syntrophy might be important in the later stage, but for the primary stage, first you have to give phenol as a sole carbon source for selection criteria. That's why option A, which is eliminating all carbon source except phenol from the media, is the correct answer. So we will move on to the next question. And the question is general staining methods do not provide information about the microbial metabolic traits. Which one of the following modifications would help us to gather this information? So the option A, fluorescent dyes that can distinguish between viable and ruptured cells. Option B, fish using DNA probes complementary to the gene of interest. Option C, cod fish using DNA probes complementary to the mRNA of interest. Codfish using DNA probes complementary to the rRNA of interest. Option E, radio labeling of gene of interest. So among this, which one of the modification would help us to gather the information? What modification would you prefer to get the microbial metabolic traits in addition to the general staining method? And your options are fluorescent dyes that can distinguish between viable and ruptured cells. Option B, the fish using DNA probes complementary to the gene of interest. Option C, the cod fish using DNA probes complementary to the mRNA of interest. Option D, the cod fish using DNA probes complementary to the rRNA of interest. And option E, radio labeling of gene of interest. You can post your answers. Your answers are option C. Okay. Option C, the fish uses DNA probes complementary to the gene of interest. So if you incorporate these changes, you could understand microbial metabolic traits. Okay. So now we will see the answer. So the correct option C is the correct option. The cod fish using the DNA probes complementary to the mRNA of interest. As we have seen Here, the microbial metabolic triad, which is we are trying to understand the function of the genes that are present in the metabolic triad, right? So you have to conduct the fish modification. That is why we are using cod type of fish, which is an advanced level of fish fluorescent in situ hybridization, where we will be doing the fish with this modification. And why we are targeting the mRNA? Because they help us to understand the specific metabolic triad that happens in different microbial community. So using rRNA binding probes instead, if you are using rRNA, it helps us to identify the taxonomic classification. That is, you will be able to identify the microorganism only. Whereas if you want to understand the microbial metabolic triads, you want to concentrate on the mRNA of interest, and you will be using more advanced fish type, which is cod fish type. And why not the other options? If you see the first option, where while fluorescent dyes can distinguish the live and dead cells, but they do not give any information about the metabolic trend. Here you could only say whether the cells are dead or viable. In option B, fish uses DNA probes complementary to the gene of interest. Yes, this statement is correct, but it can identify the presence of specific gene only, but it doesn't necessarily indicate how much level of gene expression is happening or how much metabolic activity in a metabolic community. So only fish you cannot get information about the microbial triad, but with more advanced version, which is cod fish, you can get the information out of it. And and as we have seen, while deducting the RNA, it can only tell us the taxonomic identification, that is phylogenetic identification, and not the microbial metabolic traits. And option E, radio labeling of gene of interest. What does it mean? Radio labeling means here we will use the radio isotope like carbon 13 into the DNA or RNA of specific gene, but it is not a method for studying the metabolic activity in the live population because of their radiation exposure. So that's why the option C is correct, where cod fish using the DNA probes that are complementary to the mRNA of interest. So the correct answer is option C, and we will move on to the next question. And next question is, which of the dyes or combination of dyes can be used to differentiate live and dead cells? Option A, PI, option B, AO, option C, DAPI with AO, option D, SYT09 with PI, option E, DAPI with SYT09. So, which of these dyes or combination of these dyes will be used to differentiate live and dead cells? You can post your answers. So, which of these dyes or combination of these dyes can be used to differentiate live and dead cells? And your answers are option. Okay. Your answer is, you can take time. So, which of these dyes or combination of dyes used to differentiate the live and dead cells? You can post your answers. So what is the answer? You can put your answer in the chat box. Which of the dyes or combination of dyes can be used to differentiate live and dead cells? Okay, option D, SYTO9PI. Okay, so let's see the answer now. Yes, it is correct. Option D. First, we will understand what are all these two dyes. First, SYTO9. This dye, what happens? It will penetrate in both live and dead cells, but it emits green fluorescence for the live cells. Whereas in propylium iodide, these are also fluorescent dyes. So this dye only enters into the cells which are dead and it emits red fluorescence. So it gives green and red fluorescence. If it is live cells, it will emit the green fluorescence. If it is live cells and if it is a dead cells, it will emit the red fluorescence. And if you see the options, all are like fluorescent dyes only that binds to the nucleic acid. And among them, PI is unique since it cannot penetrate the intact cell membrane and hence it can stain dead cells. But all other dyes, it can stain all the cells. And we will go through one by one. Option A, PI. PI refers to propylium iodide. So what happens? It helps us to stain the dead cells red. So it is not suitable for differentiating both live and dead cells as it is focuses only on the dead cells. And the option B, AO occurred in orange. It stains the live cells green and dead cells orange, but not commonly used for differentiating both cells. And option C, DAPI and AO. DAPI, which helps us to stain the nuclear, but this combination does not provide clear distinct between the live and dead cells. And if you see the option E, DAPI SYTO9, DAPI, as we all know, that it can stain the nuclei, and SYTO9 it can stain the all the cells. So this combination does not differentiate both live and dead cells because it is helps to stain all the cells in the nuclei. So it is also a wrong statement. So the option D, SYTO9, which helps us to stain the live cells by emitting the green fluorescence, whereas PI, which helps us to stain the dead cells by emitting the red fluorescence. So we will see the next question. Which of these habitat have the lowest fraction of unculturable microorganism? So which of these habitat has lowest fraction of culturable microorganism? Option A, seawater. Option B, freshwater. Option C, mesotrophic lake. Option D, activated sludge. And option E, soil. So among these habitat, which has the lowest fraction of culturable microorganism? So among these habitat, which has the lowest fraction of culturable microorganism? Option A, seawater. Option B, freshwater. Option C, mesotrophic lake. Option D, activated sludge. And option E, soil. You can post your answers. Answer is A. Okay. Seawater. Okay. So let's see the answer now. Option A. Yes, it is correct. Option A, seawater as the lowest fraction of culturable microorganism. First, we will understand the term fraction of culturable microorganism. So what does it refers to? It refers to the proportion of microorganism that can be grown in the laboratory setting. So now we will correlate the fraction of culturable microorganism and we will analyze the answer. First, if you see the seawater, in seawater we have very low nutrient content. Mostly mineral salts will be present in the seawater. So there may be limit of number of culturable microorganism. That is why it has very lowest fraction because it limits the number of microorganisms that are present in the seawater. And if you see the freshwater will have a moderate nutrient content. Same applies with the mesotrophic lake. Both have moderate nutrient content which allows or supports the growth of moderate number of culturable microorganisms. Whereas in the activated sludge, as the term itself denotes it's a sludge, so there will be higher concentration of organic matter that will be present. So there will be a diverse, complex microorganism will be present in the environment. So the chances of a culturable microorganism is higher in case of activated sludge. Whereas the option E, soil, and soil, we know that it is a complex and diverse ecosystem. So we could expect a large microbial population in the soil. So among these statements, seawater has the lowest fraction of culturable microorganism because of its lower nutrient content. So yes, it is correct. Option A is the correct answer. We will move on to the next question. So which one of these following gene can be used as a functional marker? So which one of the following gene can be used as a functional marker? Option A, AMOA gene. Option B, RPO BP gene. Option C, 18S rRNA. Option D, 16S rRNA. And option E, ITS sequences. So among these sequence, which can be used as a functional marker? And earlier only we have discussed taxonomic marker and functional marker. And what is their role? we have seen in the microbial community. So, which one of these following gene can be used as a functional marker? You can post your answers in the chat box. And your answers, sir? Your answers are option A. Okay. Option A, AMO, A gene. We will see the answer now. So the correct answer is yes, it is correct. Option A, AMO-A gene is the functional marker. And we will see. So AMA gene is the marker for ammonia oxidation function. So what is this gene codes for? It codes for ammonia monooxygenase enzyme. So which is the key enzyme in the nitrogen metabolism of many bacteria. So this enzyme, what it plays an important role in the process of nitrification, converting ammonia to nitrite. So this is one of the key enzyme and it is used as a functional marker for identifying ammonia oxidation function. Then, so due to its specific function and widespread presence in nitrogen cycling bacteria, AMO-A is considered as a functional marker for identifying and studying the nitrogen-related microbial community. So that's why this AMO-A gene is the marker, the two functional marker. So we will see why other options are incorrect. So if you see the RPOB gene, what it codes for? It codes for beta subunit of the polymerase, RNA polymerase enzyme. That is why RPO or RNA polymerase, which is beta subunit. So what it involves in the transcription, but it is not directly linked to the specific function activities as we seen in the AMOA gene, which play an important role in the nitrogen metabolism in many of the bacteria. And if we see C, D, E option, this is 18S rRNA, 16S rRNA, and ITS sequence, these are all taxonomic markers. So what does it tell us? 18S rRNA, it is used for identifying the eukaryotic organism. So it is not a functional marker for identifying the specific microbial group based on their unique metabolic activity. And if we see the 16S rRNA sequencing, it is used for bacterial identification. Again, it is not directly linked to the specific function as we have seen in MOA. So it is primarily used for phylogenetic analysis and determine the bacterial diversity. And if we see the ITS sequence, it is also an taxonomic marker used for fungal identification. So again, this is not a functional marker as we seen in AMOA. So among these statements, AMOA, which is the functional marker. So now we will move on to the next question, which is 16S rRNA based analysis is usually performed using the total DNA instead of total RNA because of practical limitation. Assuming total RNA could be used for this analysis, what additional information could be obtained? The question is usually for 16S rRNA, we mostly focus on DNA, not the RNA. And in the earlier lecture, we have seen why RNA is not targeted because of their instability and poor extraction efficiency. But this question telling us we will assume that RNA is being used for 16S RNA based sequencing. So what additional information can you get from this if you are using total RNA as the template? I hope you understand the question. Now I will read out the options. Option A, taxonomic identity of all the organisms. Option B, taxonomic identity of all the prokaryotic microorganisms. Option C, taxonomic identity of all the metabolically active prokaryotic microorganisms. Option D, functional capability of metabolically active prokaryotic microorganism. Option E, total metabolic capability of prokaryotic microbial community. So I will repeat the one question once again. 16S rRNA based analysis, it is usually performed using the total DNA instead of total RNA because of practical limitation. Assuming total RNA could be used for this analysis, what additional information could be obtained? You can take your time and analyze the question first and try to read the options, and you can put your answer in the chat box. And your answers are what additional information you will be getting if you are using RNA as a template. So what is the answer? Option C. Okay. What is option C? Taxonomic identity of all metabolically active prokaryotic microorganisms. Okay. So let's see the answer. Yes, it is correct. Option C, taxonomic identity of all the metabolically active prokaryotic microorganism. And why it is so? As we know, the RNA degrades quickly in the nature as they are unstable, and hence RNA-based analysis is used to get the information about the microbial and cellular state at specific point. 16S RNA-based total RNA analysis will reveal the taxonomic identity of all the actively metabolizing prokaryotic cells at the specific point. Conversely, 16S rRNA analysis from DNA includes DNA from dead cells also. So we will see why other options are not correct. If you see the taxonomic identity of all these cells, for this analysis you will be requiring 16S rRNA, which is DNA based, where you will try to identify what are all the microorganisms that are present in the sample. So it is a general state. Whereas in option B, taxonomic identity of all the prokaryotic microorganisms, here this is also a primary use of 16S rRNA, especially with prokaryotic microorganism. Whereas if you see the option D, functional capability of all metabolically active prokaryotic microorganism, here it doesn't provide any functional information. Whereas total metabolic capability of prokaryotic microbial community, so this requires functional genome analysis, not 16S or RNA sequencing. That's why with additional total RNA, we could taxonomic identity identify all metabolically active prokaryotic microorganisms. So we will move on to the next question, which is the term GC clamp is associated with which one of the following technique? This we have seen earlier. We have discussed about the GC clamp. So where we have studied the GC clamp in which technique? Option A or D or A, DGGE, option C fish, option D MLST, and option E clone library. So among this technique, the term GC is associated with which technique? You can observe the questions and take time. The term GC clamp is associated with which of the following technique? Option A. First, we will see the expansion A or D or A, which is what is it is? It is amplified ribosomal DNA restriction analysis, and DGGE, denaturing gradient gel electrophoresis. Option C, fish, fluorescent in situ hybridization. And option D, MLST, which is multi-locus sequencing typing. And option E, clone library. So among this, which is suit for GC clamp? This we have seen earlier too, where we have seen exclusively the separation based on melting profile. And your answer is option B. Okay. Option B is DGGE. Okay. Okay. Let's see the answer now. Yes, it is correct. Option B, DGE. First, we will understand what is DGE. It is denaturing gradient electrophoresis, where this GC clamp prevent the total denaturation of the sample DNA fragment. And here the separation is based on melting profile. And we have to understand what is GC clamp then. So GC clamp is nothing but it is a short sequence of nucleotide where there will be high GC content that are added to one end of the PCR primer. So that's why this technique is called denaturing, while using denaturant either by increasing the temperature or using appropriate denaturant like formaldehyde or urea, and we will run the gel electrophoresis to prevent the entire PCR product from melting completely during the electrophoresis. So this allows the better resolution of the sequence differences between the different DNA sequences. So here the term GC clamp is related to DGGE, and we will discuss why not other options. Option A, what it primarily deals with. So this technique, what it uses restriction enzyme. Option A, to digest the PCR amplified fragments, so it doesn't utilize any GC clamp. So it mainly restricts to restriction enzymes. Whereas option C, fluorescent in situ hybridization, what happens here? It is a technique that uses fluorescently labeled dye. For what purpose? It helps us to visualize both dead and live cells. That is to visualize specific DNA or RNA sequence within the cells, and this also does not involve any GC clamp. And option D, MLST, which is multi-locus sequencing typing. So what does it mean? It is molecular typing technique which uses sequence of several specific housekeeping gene to identify the bacterial isolate. So this also does not involve any GC clamp. And last one, which is clone library. What happens? The term itself tells us it is a collection of cloned DNA fragment that are inserted into the vector. So here also it doesn't correlate with the GC clamp. So the only term that GC clamp is associated with is DGGE, which is denaturing gradient gel electrophoresis. So yes, the answer is correct, DGGE. So we will move on to the last MCQ, which is comparison of restriction site location within the same target gene is performed using, so here restriction site location. So if it is restriction site, what will you perform? Option A or D or A. Option B, DGGE. Option C, fish fluorescent in situ hybridization. And option D, MLST. And option E, clone library. So I will repeat the question. Comparison of restriction site location within same target gene is performed during. You can type your answer now. You can take your time. Comparison of restriction site location within same target gene. This also we have studied in today's lecture. And your answers are A. Option A. Okay. Option A or D or A, which is amplified ribosomal DNA restriction analysis. So we will see the answer. Yes, the correct answer is A or D or A. The term itself suggests that it is amplified ribosomal RNA restriction analysis. What he had done, so this subjects to amplicons of 16S rRNA gene to restriction digestion. So what happens here after extracting the DNA out of it and doing PCR amplification, we will select restriction enzymes in order to cut the DNA at the specific site in order to compare the lengths of resulting fragment. So this technique often involves comparing the DNA fragments generated by restriction enzymes cutting at the specific site within the target gene. as like 16S or RNA gene. So the option A is correct, whereas the option B, what it tells, it separates the DNA fragment based on their size and melting point. So it does not separate based on their restriction site location. So though it can analyze the DNA fragment generated by the restriction enzymes, the primary focus of DGG is melting point differences rather than the restriction site. That's why option B is incorrect. Whereas if we see the fish technique, as we already know, it uses fluorescently labeled probe to visualize the specific DNA or RNA within the cell, and it helps us to compare the live and dead cells. So it is not associated with any restriction site location. Whereas if you see the option D, MLST, as we have seen, it is used to compare the sequence of several housekeeping genes to identify the bacterial strains. So it is also not correlating with restriction site location. And if you see the clone library, so it is a collection of clone DNA fragments that are inserted into the vector. So it is also not directly comparing the restriction site location within the single gene. So among these various techniques, the only technique amplified RNA ribosomal DNA restriction analysis uses the restriction site location to identify the microbial organism. So, so this is all for the today's class. And once again, thank you for patiently listening to my lectures. And if you still have any doubts, you can mail me at ajitvelu284@gmail.com, or you can post your queries in the discussion forum so that our team will try to resolve as much as query possible. And I hope you are all doing your assignments on time because it plays an important proportion for your final evaluation. So try to finish your assignments on time. And if you have any doubts, you can put your queries in the discussion forum. And if you have missed out any of my previous lecture, you can go to my YouTube channel, which is Ajitkumar_V, and you can watch it any number of time if you have any doubts on the lectures. And you can also use the lecture videos and the PPTs that are available in the NPTEL Environmental Biotechnology course, where you can access these videos as well as the PPT in the recording session. So this is all for today's class. And if you still have any doubts, you can enter your queries in the chat box. And in today's class, we have seen the culture independent techniques, and we have seen different techniques and how it is important for next gen sequencing technology. And we have seen their evolution, and we have seen different omics approaches like metagenomics, metatranscriptomics, metabolomics, and metaproteomics. So these are all more advanced omic approaches that are used in parallel in order to understand the microbial community. So stay tuned for the next lecture also. Next lecture, we will be discussing the more important topic, that is the application of environmental biotechnology, where one of the primary application is bioremediation. And we will see the importance of bioremediation from the next lectures, and try to attend the lectures as possible because here we will be discussing the various MCQs or sample MCQs, which helps you to understand more concept efficiently. So try to attend the lectures and get benefit out of it. And still, if you have any doubts in the lectures, you can revisit the professor lectures also, and also our team members lectures. So you will get a clear picture out of it. So that's all for today's. Have a great day and have a great weekend. So still, you have any doubts, or we can wind up the session? Can we wind up the session? Okay, thank you, Ashwini. So try to attend as much as possible and try your friends also to join in these lectures so that they can also benefit out of it. So thank you once again. Thank you once again, all. Thank you, Ashwini. So thank you once again, all. Try to attend the lectures as much as possible and finish the assignments on time because it holds an weightage to your final examination. So thank you all. Have a great day. We will close this session. You can leave now.